Buenos días. Good morning, everyone.
Good morning. So I'm waiting until 7 a.m. to present you formally. Okay, good morning, everyone, and thank you for coming to this new keynote talk. Today we have the pleasure to present Professor Megan Frederickson. Professor Megan did her PhD in biological science at Stanford University. She was a junior fellow in Harvard Society of Fellows. She was an assistant professor and currently is an associate professor in the Department of Ecology and Evolutionary Biology at University of Toronto, as well as a Radcliffe Fellow at Harvard University. Professor Megan's research interests are around evolutionary stability of mutualism, evolution of cooperation in ants, consequences of mutualisms for ecological communities, and ecological genetics of mutualisms. Professor Megan, you are very welcome. You can start.
Thank you for the introduction. Gracias a todos por venir a mi charla. Lo voy a dar en inglés, pero quería darles la bienvenida en español y decir gracias a los organizadores por invitarme a dar esta charla en el simposio. Estoy obviamente en mi casa en Toronto. Es un nuevo mundo en que tenemos esas conferencias virtuales. Pero estoy muy feliz de estar hablando a ustedes hoy día. Okay, now I have to see if I can share my slides. Can you see my slides?
Yes, perfectly.
Okay, great. And you can still see me because I can't see anything but my slides.
Yes, we can see you.
Fabulous. All right. My talk today is entitled How Mutualism Evolves Between Ants and Plants. And this is one of my favorite mutualisms. This is an ant that's visiting an extrafloral nectary. This is actually a painting that one of the undergraduate students who works in my lab did a couple of years ago. And in my lab at the University of Toronto, we study ecology and evolution of mutualisms in the broad sense. So I'm going to talk about my research on ant-plant mutualisms today, but we actually work on a wide variety of plant, animal, and host microbe mutualisms. So I got my start working on ant-plant symbioses in tropical rainforests, and I am going to talk about ant-plant research today. But as my lab has grown, we've also dabbled in pollination biology. I've had a big project looking at seed dispersal by ants. We also now do quite a lot of work on host microbe interactions, including between duckweeds and their microbiomes, and between legumes and rhizobia. And I've collaborated on some work looking at the microbiomes of ants as well. But what unites all of these different themes is really an interest in understanding how cooperation evolves between species. So many myrmecologists are interested in the evolution of cooperation. One big reason a lot of people study ants, of course, is to understand how it is they came to be eusocial and how it is that they evolved to live in colonies with a queen and sterile workers. And I'm very interested in social evolution as well, but in my lab, we principally work on mutualism evolution, so how cooperation evolves between species, not within a species. And one of the reasons that this is interesting to us is because unlike when we think about cooperation within a species, which may evolve through kin selection, cooperation between species happens between partners that don't share genes, so it precludes inclusive fitness benefits. I'm very interested in, nonetheless, how cooperation evolves between species, even when they don't share genes. And the work I'm going to tell you about today was carried out by my amazing graduate students and postdoc, and you can see their photos here in this slide. And I'm going to tell you about two projects, or I'm hoping I'm going to have time to tell you about two projects today. The first one is kind of a microevolutionary project, and the second one is a macroevolutionary project. So in the first project, I'm going to talk about recent work we've been doing that's attempting to unravel the molecular basis for ant cooperation with plants. And in the second part, I'm going to talk about mutualism, evolution, and ant diversification at a macroevolutionary scale. So in this first part of my talk, I need to introduce to you a little bit about the study system and a little bit about defensive ant-plant mutualisms in general, because that's what's the focus of this first part of my talk. So in many tropical plants, we have these myrmecophytic lineages that host ants that defend them against herbivores. And the way this works is we imagine we have a host plant like the one shown in this cartoon. And the big problem this host plant faces is that it's eaten a lot by herbivores like this grasshopper. But this host plant has evolved ant demetia, which are hollow, swollen structures that house ants. In this case, it's a hollow, swollen stem that houses ants. And these ants act as effective bodyguards and provide protection against herbivores to plants. So the bodyguard ants attack the herbivores, and this reduces the amount of herbivory on host plants. It's beneficial to plants because they have less herbivory, and it's beneficial to the ants because they get these nest sites and often food as well from their host plants. And the specific ant plant or myrmecophyte that I'm going to talk about today is Cordia nodosa, which is shown here in this photo. And it houses quite a few different species of ants, but I'm going to focus on just one of them today, which is this ant, Allomerus octoarticulatus. And Cordia nodosa is a plant that makes a lot of these hollow, swollen stem demetia. So there's one on every node that it grows on this tree, and it's a kind of a short-statured tree, so it grows to maybe a few meters in height. So it's an understory tree that grows throughout the Amazonian rainforest, and I've studied it principally in Peru. And when it has ants, in particular when it has Allomerus octoarticulatus ants or sometimes some other ant species, it is well defended against herbivores, and you can see that it suffers very little damage to its leaves from herbivores, like in the left-hand side. And when it is unoccupied, when it doesn't have ants, we frequently see that it's very heavily eaten by herbivores. So that's immediate evidence that these ants are beneficial to the plant. And as I said, I've studied this mutualism, well, really, I've studied this mutualism throughout the Peruvian Amazon, but I'm going to talk about work today that I did at the Los Amigos Biological Station in Madrillos, Peru. You can see where it is on the map. It's a beautiful field site. I rather wish I were there right now, but hopefully I will be able to go back in the not too distant future. So in animals, in general, cooperation depends largely on behavior. And this is true both in terms of social behavior or cooperative behavior within a species, so the behaviors that ants have inside in their colonies or in their nests. And it's also true in plant-animal interactions. In most plant-animal interactions, the cooperation depends on behaviors of those animals. And that's true in pollination, it's true in seed dispersal, and it's true in these kinds of ant-plant protective mutualisms that I just described to you. As a result, to understand the evolution of cooperation in animals, we need to understand the genetic and molecular bases of cooperative behaviors. I think it's important not to treat cooperation as kind of a special trait, and instead we need to treat cooperation as a trait, much like any other, as if it were, a morphological or physiological or any other kind of behavioral trait. Because if we treat it like a trait like any other, then we can use it to understand how cooperation evolves. And to do this, one thing that we need to do is to consistently measure phenotypes like cooperative animal behaviors across many individuals, or in the case of ants, many colonies, so that then we can associate phenotypic variation with molecular and genetic variation in order to understand how molecules and genes give rise to these phenotypes of interest, in this case, cooperative animal behaviors. And in animal mutualists in particular, cooperative behavior is generally foraging behavior. So pollinators are really just foraging for nectar and pollen when they visit flowers. Seed dispersers are really foraging for fruit or elastomes in the case of ants when they pick up and move seeds. And even in protective ant-plant mutualisms, ants are often foraging for insect prey or sometimes for extrafloral nectar or other plant-provided food sources as they're defending plants against herbivores. And that's going to be important in a moment. So my approach to trying to get at the genetic or molecular basis of ant bodyguard behavior in an ant-plant mutualism has involved trying to consistently measure variation in this behavior across ant colonies. So I'm going to go into some detail about how we have been measuring how good ant colonies are at defending their plants against herbivores. And so we do that in a short bioassay that works as follows. So here again is our cartoon of a domatium of this ant plant, Cordia nodosa, and you can see it has this hollow stem domatium that ants are living in. And there might be some ants that are out patrolling the leaves, waiting for herbivores to attack it. And so what we have been doing is we catch some herbivores, generally grasshoppers, at our field site, and we pin them to leaves, and then we just measure how many ants attack the herbivore over a period of 5 minutes. So it's not a very long bioassay. We just count the number of ants in contact with the bait insect every minute for 5 minutes. In general, when we pin the grasshopper to the plant, it starts off with no ants, and then gradually ants arrive, and then sometimes it kind of levels off as some number of ants are then busy attacking this herbivore and providing this bodyguard service to their host plant. And after we're done with this bioassay, where we've measured how active the ant colony is at defending its plant against herbivores, we collect the bait insect with the ants attached, carefully remove very quickly the ants, store them in RNA later for later sequencing, and also store the grasshopper in ethanol for identification and to measure its size, for example, in case bait size is an important variable we need to consider for bodyguard behavior. And on these plants that we've measured the activity of these ants as bodyguards, we've also measured the amount of herbivory the plant receives, usually over a period of 2 to 3 weeks. And we've done that just in the standard way that plant ecologists do, which is just to mark some leaves and measure the amount of leaf area that they have lost to herbivores at some initial time, and then measure it again 2 to 3 weeks later. So this is what these bioassays actually look like when we carry them out in the field in the Peruvian Amazon. This is kind of a moderately active Allomeris octoarticulatus colony. You can see that the ants are attacking the legs of this herbivore. And this is a more active Allomeris octoarticulatus colony that's really gone after this bait insect quite aggressively. And we see this very commonly, that this ant species shows a lot of variation in bodyguard behavior, where some ant colonies are extremely aggressive and provide really good bodyguard service to their plants, and some ant colonies are not so aggressive. And you can really see that in these data. So here is what these bioassays look like in aggregate across lots and lots of different ant colonies. So we've measured, at this point, ant activity across at least a few hundred ant colonies. And on the left-hand side, you can see the number of ants attacking the bait insect on the y-axis across time, so the 5 minutes of each bioassay, with the solid blue line showing kind of what the average ant colony does, but what those light gray lines are each a distinct ant colony. And what I hope you can see is that there's a lot of variation in this behavior. So some ant colonies have high numbers of ants that recruit to the bait insect, and some ant colonies have very few or even zero that basically don't respond at all. And this variation in our very sort of quick and dirty bioassay that only lasts 5 minutes still tells us something important because it's negatively correlated with the amount of herbivory on the attached leaves. So on the right-hand panel is this bodyguard activity on the x-axis. It's actually the square root of the average number of ants attacking the herbivore over the 5 minutes of the bioassay. And herbivory, just the rank of herbivory. So leaves that had very little herbivory have a low rank, and leaves that had very high herbivory have a high rank on the y-axis of this figure. And so you can see that ant colonies that have more active bodyguards experience less herbivory. All right, so we were interested in figuring out what explains some of this natural variation in this behavior that we see. We know that this behavior is important to plants because I just told you that it's correlated with herbivory in plants. And so why is it that some ant colonies are really good at this and some ant colonies are not? And of course, there can be both environmental and genetic influences, or environmental and molecular influences on these behaviors. And I'm going to get back to the issue of environmental influences in a little bit, but I'm going to start off by thinking about the potential influences of molecular mechanisms. And I happen to have an office right next door to Marla Sokolowski at the University of Toronto. And Marla Sokolowski and her lab over many years have been working on behavioral genetics, and in particular, the role of a gene known as the foraging gene in shaping animal behaviors, principally in fruit flies, but also in other animals. And the foraging gene encodes a cGMP-dependent protein kinase named PKG. And Marla Sokolowski's early work on the foraging gene in fruit flies discovered that there are these natural allelic variants of this gene that are called rovers and sitters, and that they show different foraging behaviors. So in the top right part of this slide, you'll see a figure from one of her papers, and what it's representing is a behavioral assay in which the larvae of fruit flies are foraging on yeast patches in agar plates. So these larvae are eating yeast. And there are these two kind of yeast patches on each plate, which are shown in the darker beige color across these agar plates, which are kind of in a more lighter yellow color. And the flies that are genetically rovers, as their name implies, move a lot between food patches in search of food. But these flies that are genetically sitters, as their name implies, just stay in their food patch and eat. And so this was one of the first demonstrations that there can be a gene that has a large effect on an animal behavior, in this case, larval foraging behavior. But since then, there's been a lot of work done showing that other animals, including bees and ants, in other animals, including in bees and ants, this gene can have important impacts on behavior. And in particular, it's often the case that the expression of the foraging gene changes as workers in bee or ant colonies transition among tasks. So for example, as they go from being brood care workers inside a nest to foraging outside a nest for food. And so, as I said, I have an office right next to Marla Sokolowski's office, and I got interested talking to Marla and hearing about the work of her students. I got interested in knowing whether the foraging gene might also be important in ant plant mutualisms, and whether it would give us a bit of a window into understanding the molecular basis of ant bodyguard behavior. So is there a role for this highly conserved gene that's known to have impacts on other animal behaviors? Is there a role for it in an ant-plant mutualism? And because this gene encodes a cGMP-dependent protein kinase, PKG, it's possible to actually pharmacologically manipulate the activity of this gene product using this activator called 8-bromo-cGMP. You don't need to remember its name, you just need to remember that it's a PKG activator. Basically what it is a synthetic form of cGMP that activates the protein that's encoded by this gene and therefore makes the animal behave as if it were expressing higher amounts of this gene. So in the field in Peru, we fed this PKG activator to ant colonies for 2 weeks, and before and after, we measured ant bodyguard behavior. So we measured ant aggression to grasshoppers, as I explained in that bioassay, as well as the damage they experienced to young leaves, so as well as herbivory. So here are ants at one of the tubes that was providing either this PKG activator or just a control. It was dissolved in sugar water, so the control was just sugar water. And these were the results of that experiment. So when we treated these ant colonies with this PKG activator, they really did start to attack these grasshoppers more in our bioassay compared to ants that just got the control treatment, which again was just sugar water. And that's what's shown on the left-hand side. And we also found that this treatment affected herbivory on plant leaves, so that the ant colonies that received the activator had leaves that experienced less herbivory over the course of this experiment than the control colonies that just got sugar water. So this suggests that PKG activation in Allomeris octoarticulatus can make ants better mutualists. They become more aggressive, and it has these knock-on effects on how much herbivory their plants get. All right, so that was a kind of an interesting first attempt to understand something about the molecular basis of bodyguard behavior in Allomeris octoarticulatus. But we were also interested in looking not just at a single candidate gene like foraging, but really looking across the transcriptome to see how gene expression was correlated with this behavior of interest. So again, we measured ant bodyguard behavior in these bioassays. We also measured a whole bunch of potential environmental influences on bodyguard behavior. So temperature and colony size and plant size and the time of day that we did the bioassay and what bait insect species we used and the size of the insect and so on. And to a first approximation, none of these had consistent effects on the behavior. So none of these variables consistently predicted ant bodyguard behavior. So we took our ants that we collected when doing these bioassays, and we sent 20 ant samples for RNA sequencing, and we chose samples for RNA sequencing that were from the high and low end of the distribution of bodyguard behavior. So we chose basically 7 colonies that had very low levels of aggression towards these herbivores, and 7 colonies that had very high levels of aggression towards these herbivores, and we did RNA-seq on these bodyguards from these colonies. And in a few of these colonies, we'd actually also opened up Demetia and collected workers that were caring for brood inside. So we also had some RNA-seq data on matched samples of brood care workers for a few colonies. And I have some very preliminary results to share with you from our gene expression study. I'll say that this is extremely preliminary. It's kind of hot off the press, and it may change still, so we haven't submitted this work for publication or anything. But basically, on the left-hand side, you're looking at an analysis of what genes in the Allomerus octoarticulatus transcriptome were differentially expressed between ant colonies that had high levels of aggression to these bait insects, so that were really good bodyguards, and ant colonies that were not very aggressive towards herbivores. And the red dots are genes that had significantly different expression between those two groups of ant colonies, and that there are 32 of those genes, meaning that we have identified some genes that in this very preliminary analysis are potentially related to bodyguard behavior. Our comparison of bodyguards and brood care workers in these ant colonies yielded a much larger number of genes, which is not really surprising given how much we already know about gene expression differences between brood care workers and foragers, for example, in other kinds of ants. And this analysis, which is on the right-hand part of this slide, showed that there were 632 genes differentially expressed between bodyguards that were outside the domatium attacking herbivores and brood carrier workers that were inside the domatium tending to brood. And we've been starting to work on functionally what these genes are, and I do have some preliminary results, but I don't have enough faith that our functional identifications of what these genes are correct, so I'm not going to show them to you at the moment, but I'm happy to talk to anyone who'd be interested to know more about that. So in conclusion to this part of my talk, the take-home messages are that bodyguard behavior, and thus the quality of ants as mutualists, is linked to gene expression. I did want to say, though, that gene expression is still just a phenotype. It's not a genotype. So we still have a lot more work to do to understand what genes actually encode ant traits that affect plants. And hopefully that is something we'll be able to do as we get more information on what genes at least are differentially expressed between ants doing different behaviors. And herbivory on ant plants is likely jointly influenced by both ant and plant genomes. If you like, it's a joint phenotype of both ants and plants in a system like this. And I've just co-authored a paper that's been accepted at Proceedings of the Royal Society B, looking at, or that modeled the evolution of joint phenotypes that are determined by both host and symbiont genomes. And I think there's a lot of interesting things that we could think about in terms of how those kinds of traits evolve. Okay, so the second part of my talk, assuming I'm still doing okay on time, is on mutualism evolution and ant diversification at a macroevolutionary scale. So now we're going to think about ant evolution at a big scale. And in fact, I'm going to take a step further back than that and think about sort of the evolution of life on Earth in general. It's the biggest scale possible. So I've always really liked this quote from Lynn Margulis and Dorian Sagan, who said in a book, "Life did not take over the globe by combat, but by networking." And of course, they were thinking about the evolution of the eukaryotic cell via endosymbiosis. But I've always wondered how generally true this might be. How often is it the case that symbiosis or mutualism leads to ecological and potentially evolutionary success of lineages? So is mutualism a key innovation? It has been argued to be a key innovation in quite a number of different cases. So of course, as Margulis was hinting, we think it was critical to the evolution of the eukaryotic cell via endosymbiosis that then led to the radiation of eukaryotes around the globe today. It's also been argued to be important for plant invasion of dry land, which was thought in some cases to, or it's thought by some people to have been enabled by mycorrhizal fungi. And we also often think about whether pollinator-driven or pollinator shifts among different lineages of angiosperms has led to the radiation of the angiosperms over evolutionary time. So these are all cases in which mutualism has been argued to be a key innovation. But when we think about plants and insects, and when we think about coevolution, of course, the dominant hypothesis has sort of been the opposite. We know from Ehrlich and Raven's famous 1964 paper on coevolution, they gave us this big idea that plant diversity and insect diversity around the globe has really been due to escape and radiate coevolution, that plants have diversified by evolving a novel defense that allowed them to escape their herbivores and diversify free from herbivores for some period of time. And then eventually insect herbivores evolved to be able to eat some new plants, and then they've diversified thanks to the sort of new ecological opportunity that's opened up by being able to eat a new food source. So it's thought that antagonisms and not mutualisms have been really important to the evolution of plant and insect diversity. But in the study I'm about to tell you about, my lab was interested in understanding whether not escape and radiate coevolution, but what we termed cooperate and radiate coevolution might also be important for diversification, especially of plants and insects, or in this case, especially of plants and ants. So this is a summary slide showing some recent estimates for the number of ant and plant taxa that engage in ant-plant mutualisms. And we know that there are roughly 11,000 plant species that have eliosomes. So they have these lipid-rich appendages, like the one you can see on the slide, that attract ants for seed dispersal. And there are at least 309 different ant species that are attracted to eliosomes and disperse seeds. There are at least 681 plant species that have ant domatia, that have these hollow structures that house ants. And they are, and there's at least 58 ant species that nest inside domatia, and that there are at least 3,941 plant species that make extrafloral nectaries, so glands outside flowers that secrete sugar-rich nectar that attract ants that provide defensive service. And there are at least 122 different ant species that visit extrafloral nectaries. And some previous work before I started working on how mutualism affects ant diversification, some previous work had looked at how ant-plant mutualisms affect plant diversification. The top left phylogeny is from a study by Marjorie Weber and Anurag Agarwal from PNAS in 2014, in which they looked at how the evolution of extrafloral nectaries in plants affects plant diversification. And they found that lineages that have extrafloral nectaries have diversified faster than lineages that don't. And an older study also found a similar thing for eliosomes, that plant lineages that have eliosomes are more diverse than their sister taxa that do not have eliosomes, suggesting that these traits have been important for plant diversification. But I was interested to know whether the reverse is true. Does mutualism evolution affect ant diversification? And to figure this out, we took kind of a novel approach and used some computational tools to do a text mining project. So we actually text mined over 89,000 abstracts of papers that are about ants, and we got them from two sources. One was the Formis database, which has all known ant literature up to, I think it's 1996. And the other was the Springer family of journals, which includes, for example, Ecologia and Insect Socio and lots of other journals with substantial ant content. And we did something very simple. We just took a list of known ant names. We searched for each ant name in each abstract, and then we also developed a list of trait terms that indicated whether those ants might associate with plants. And the trait terms were things like elizome, extrafloral nectar, Demacia, etc. And we counted how many times an ant name co-occurred in an abstract with one of these trait terms. And then we manually checked ant names that appeared in only 5 or fewer abstracts to kind of ground truth our data set. So this is what the data looked like. We turned it into binary trait data that we generated through this text mining algorithm. I took a chunk out of the data set. This is just for Ectotoma. And basically, it turned it into a series of zeros and ones, where a 0 means that we found that ant name in abstracts, but it was never in association with any of our trait terms that suggest it interacts mutualistically with plants. And a 1 means that that ant name was associated, in this case, with the trait term extrafloral nectar or extrafloral nectary. And you can see that this at least sometimes correctly captures trait data because, for example, our text mining found that Ectotoma iridum associates with EFNs. And here's a photo from Alex Wild's beautiful website showing Ectotoma iridum visiting an EFN, and the same thing for Ectotoma tuberculatum. And then we plotted these trait data across an ant phylogeny that was provided to us by Matt Nelson and Corey Moreau. And here I'm showing you the trait data collapsed for each ant genus. We actually did our analyses at the species level, but when I show you the species level tree, you can't really see anything. So I'm going to show you the genus level tree just to be able to show you a little bit better how this works. So now around the tips of this tree, the boxes indicate whether the lineage has or doesn't have an association with plants that bear Demacia in pink, with food bodies in yellow, with extrafloral nectaries in kind of a bluish purple color, with whether the ant lineage disperses seeds in orange, or does any of these things, which is shown in green. So is a plant mutualist in general. And around the bars that are around the edges of the circle indicate the number of species in each genus, and the number of ant species in each genus. And you can see that it does kind of, tend to line up that genera with more species are more likely to be plant mutualists. But we did our analyses, as I said, at this species level scale with roughly 800 tips in this tree that we had trait data for. And then we modeled the association between being a plant mutualist and diversification rate. So the net difference between speciation rate and extinction rate across the phylogeny. And we did this in 3 different ways. The first way is in a BISI model, so a binary state speciation and extinction model. And when we did this, we found that the plant mutualists, so ant lineages that have evolved a mutualism with plants, had a higher diversification rate than ant lineages that had not. And in a second modeling approach called BAM, or the Bayesian analysis of macroevolutionary mixtures model, we also found the same thing where lineages that were plant mutualists diversified faster than lineages that were not. But for any of you who know this literature, and specifically the literature on trait-dependent diversification, or these state-dependent speciation and extinction models, you'll know that there has been a lot of debate over the validity of some of these phylogenetic comparative methods. And there's a fear that they can produce kind of spurious results. And so we also did a third kind of model called HISI, which is a hidden state speciation and extinction model. And what HISI does differently is it allows there to be some other hidden state that you don't have any information about that might be affecting diversification rate, And then you can ask whether your trait of interest has an effect over and above this background heterogeneity in evolutionary rates. And when we do that, we find the exact opposite result. We find that plant mutualists are diversifying more slowly than non-mutualists. Although interestingly, mutualism was far more likely to evolve in clades that were already rapidly diversifying in this Hissey model. So the conclusions from this part of my talk are that Bissey and BAM models really found support for our hypothesis of cooperate and radiate coevolution between ants and plants. But Hissey is really more consistent with what we called a radiate then cooperate scenario, where mutualisms evolved more frequently in lineages that were already rapidly diversifying, probably for some other reason, but then subsequently slowed diversification. So I'm going to stop there and acknowledge the people who are important for doing this work and for my funders. And I'm happy to take questions, but in order to do so, I think I have to exit out of my presentation because I can't see anything other than my slides. All right.
Oh, thank you very much for your talk. I'm going to ask you some questions from the public. So Professor Fernando Fernandez says, congratulations for the presentation. There are plans for study the internal phylogeny of Alomerus. Some observations by Orivel in French Guiana suggest that Octorticulatus comprises at least 2 species, each one associated to different plant genus.
I think we're going to have somebody moderate the questions, and I'm very happy to answer them if anyone wants to know more.
Did you hear my question?
Yes.
So I was saying that we have 3 questions. So the first one is from Professor Fernando Fernandez. He says, congratulations for the presentation. There are plans for the study the internal phylogeny of Alomerus. Some observations by Orivel in French Guiana suggest that Octorticulatus comprises at least 2 species, each one associated to different plant genus.
Yes, I think it would, I am excited to see what others what results are generated by a better phylogeny for Alomerus. I think that at my field site in Peru, and in Cordia nodosa, at the one field site in Peru, given the genetic data we have, I'm pretty sure all of our samples are from one species. But it's true that I have wondered, looking across the range and also across some of the other plant species that this ant has been recorded from elsewhere in its range, I wouldn't be surprised if it is more than one species in the end. But in our one site, where all of our samples are from Cordia nodosa, I'm reasonably confident they're all the same species based on our genetic data, although we still have more work to do.
Great. We have another question from Felix Rosemek. He says, very interesting the effect of the foraging gene. Any clue whether it increases aggression toward herbivores or is used a probabilistic effect? More foraging, more ants finding the herbivore, more attacks?
We don't really know. That's a very good question about what exactly it's doing. In other work, so outside of Alomerus octorticulatus, it has been suggested that foraging is kind of an overall activity level gene, that it just makes things move more and be more active in general. But in ants, there's only a handful of studies that have looked at it. And interestingly, they haven't all found the same pattern, where it's not always the case that, for example, foragers express higher levels of foraging than brood care workers. People have found the reverse. So I'd say it's early days yet for knowing mechanistically what it's doing. A very good question.
Great. Andres Morales asked, great presentation. Could there be other uses for the treatment for that gene expression, maybe agriculture?
Oh, yeah. It's a good question. If we knew more mechanistically about the molecules involved in making ants really great anti-herbivore defenses, could we deploy them in an applied setting like agriculture? I don't know. I think we're a long way away from knowing enough to say anything, although I have enjoyed reading other work on ants in as defenders in in agroecosystems. So for example, in coffee over the years. So I have wondered, but at the moment, my goal has really been to understand it at a more sort of fundamental, curiosity-driven level.
Great, thanks. And our last question is, awesome and very interesting presentation. What possible hypotheses do you have for the radiate then corporate model from Giampiero Fiorentino?
Can you say that again? The last part?
Oh, yes. What possible hypotheses do you have for the radiate then corporate model?
Oh, so we were somewhat surprised to find that our data is most consistent with a model in which ants are there is already this heterogeneity in diversification rates across ant lineages. And then there are some parts of the ant phylogeny that are just diversifying very fast. And then mutualism evolution seems to slow it down. And I guess one simple thing that could be going on, for example, is if that is due to biogeographic effects. So maybe ant lineages are diversifying faster in the tropics or, in some particular biogeographic regions. And then mutualism evolution is sort of acting on top of that to actually slow it down. I would say, though, that we also, I sort of feel like I answered all of these questions in the same way, which was to say we need to know a lot more. Which I would say, though, that we have so many open questions about ant diversification, especially across all ants, because the tree that I modeled this in had 800 tips out of all ants, which are, 15,000 to 20,000 total species. So it's still grossly undersampled. And I think until we get sort of better representation, a more exhaustive phylogeny and a better trait data set, we could potentially see very different results. But what I hope that text mining project suggested was that some of these, for example, text mining methods might help us get data from a large chunk of the literature. It might be an efficient way to gather trait data from the literature in general. And I guess I'll end on saying one of the reasons I like that text mining study is that every single sort of case study where somebody made very detailed observations about what one particular ant species was doing out there in nature then becomes a data point in our, it becomes a valuable data point in our bigger study of patterns across the ant phylogeny. And so we also need everybody to be going out there and making these detailed sort of natural history and behavior and ecology studies of ants in their natural world, if we're ever going to get to the point where we have enough data to answer this question well.
Great. Megan, Frederickson, I appreciate your time. So we have to move on to the other talks. Thank you very much.
Thank you, Megan. Amazing work.
El conocimiento se fortalece cuando los nodos logran interconectarse. Bienvenidos al primer Simposio Iberoamericano de Mirmecología, versión en línea. Para más información, visita nuestra página web o comunícate a nuestro correo electrónico.